organizers for having me, and thank you for staying. I think it's a benefit of speaking before Maha. And so I will tell you about a particular system where cells come together and try to solve real-world problem, meaning defending the host, like us, against unknown world and changing one. So it's a crowded slide, but I'm trying to show a particular type of immune cell. So we heard of T cells, which are killers or helpers, which basically make a binary decision. But for B cells, it's learning lifelong, and they act in this kind of cell clusters. So here, the green things are the B cells. So they form these very dynamic clusters of cell population that compete fiercely for a limited amount of stimuli, which chase back to the molecular scale. Here is a protein on the surface of HIV virus. And it's in complex with one arm of the Y-shaped molecule, which is the antibody, which are basically the surface receptor of the B cells in secret form. So on the other hand, all this kind of dynamic population will form ensemble in our lymph nodes. So they come when the stimuli arise. And this is a stunning picture from Gabriel Victoria's lab at Rockefeller. So you can imagine these clusters can communicate and the pathway through memory cells that can migrate and reinvade some ongoing populations to basically improve the global selection pressure for a stronger response. So I'm interested in this system. One particular reason is evolving, and this particular evolutionary process that takes place in weeks, hopefully, is named affinity maturation, standing for an improving collective affinity improvement in the cell population. So if we think about the immune system as a problem solver, the task is both complex and changing. And the difference between this and the usual circumstance, I will jump to this one first, where people think about fluctuating environment, how to survive it, is mostly concentrating on some learnable statistics of the environment, meaning some recurring statistics can be extracted by the cell population, for example, by doing the back hedging strategy, by diversifying a population such that at each point in time, there's always a subpopulation that can respond properly. And you can imagine there are other mechanisms to survive. For example, just sense and respond. But the cost is high when the environmental changes is infrequent enough. Right? So, and Ido Kuso and Stan Leibler have thought about this in a phenotypic space. So this is actually an analogy with heteropolymers that will localize toward the interface between two kinds of environments that switch in time if it's composed of two kinds of monomers that favor each of them. So there's a dynamic localization transition. But these are all occurring in the phenotypic space. And if we want to understand how the immune cells evolve useful antibodies to basically try to extrapolate what's learned in the future, in the past toward the future, they need to generalize by learning from maybe a small number of examples. So I go backward a little bit. So I just want to give two examples of who are generalists. So we use the term in the molecular sense. So that means, on one hand, this is a desirable generalist, which is called broadly neutralizing antibodies. So they were discovered over the past decade as a very desirable protection for a broad range of variants of viruses, such as the rapidly evolving ones, like HIV, the notorious one, which we don't have a vaccine at all. So here I'm showing the dendrogram. So all these different branches are distinct HIV strains. So over time, the reddening of this dendrogram indicates that the coverage of response by this single antibody is increasing. So that means it can gain about 90% of coverage of all the known circulating strain over a course of 5 to 6 years in this individual that's tracked long enough. On the other hand, the multi-drug resistant bacteria is another example of generalists. So they have no trouble emerging. And they emerge quickly and are abundant. But in contrast, these desirable broad antibodies are really reluctant to emerge. So in these patients, they emerge after years of infection in tiny amount. We don't really understand the reason of the reluctance. And also, without that, we won't be able to design the examples they can learn from. Okay. So this is the question I'm trying to answer. How does evolution discover these rare solutions, given that they are really easily outcompeted by specialists in a constant environment? Okay. Yeah, so here are the people who are involved in this work. So Lei was a postdoc fellow at UCLA who's now starting his own group. And Jiming is a graduate student in my group. And Kyle just joined and starting doing some follow-up work. And we benefit a lot from talking to Arvind, who was thinking about similar problem, but in circadian clock or folding sheets, just different contexts. But the same theme is how to learn from a few examples about something not seen before. Okay. So one thing, yes, so I told you one thing special about immune system is the statistics of the environment is not known. So we can't really repeat or present the recurring patterns so that the system can extract it. Because we need to first figure out what features are recognized or what are the common features that we need to encode into the examples. And the second challenge is the map from the sequence to function is not available, meaning so for those phenotypic description, the map will be underlying it. But here we have to work from the genotypic space. So this rapidly advancing field of deep mutational scanning technique is pushing toward that direction, but so far it can only do a very small chunk of the binding domain. And maybe for the antibiotic resistance case, it's more accessible because the binding region is relatively small. But for antibody, usually it can be easily 20 to 30 amino acids involved. So it's still a challenge. But some preliminary study along this direction in Justin Kinney's lab at Cold Spring Harbor shows that epistasis, meaning the interaction between mutations, is important. So we can't really use additive model as we wanted. So we have to think about it. And the environments in those phenotypic switching contexts are assumed to be random and uncorrelated. Right? There are some basically independent challenges presented in time. But at least for HIV case, these rapidly mutating viruses are producing correlated example of itself. And here I'm showing the binding interface, basically where the antibody can step on. So the red indicates the more conserved region, where mutation is more costly to the virus. So you notice the major binding sites are basically a mix of blue and red, meaning the antibody can't avoid stepping on variable parts if it wants to target the conserved. So if we translate that to the binding landscape, in a way, the mapping will be correlated over time. So that's another challenge we need to account for. So any kind of representation will be wrong on a 2D plane, but this is helping us to imagine how we can imagine the organization of states in the sequence space. So we're interested in how the specialists and generalists are organized. So we start with something that could be wrong, but at least we start somewhere. So we are thinking about two environments. So each antigen, meaning the part of the virus that induces immune recognition, will present a landscape. Right? Each coordinate corresponds to a particular sequence of antibody, right? So we can have correlated one over time, and the simplest temporal procedure is cycling them. Okay. So how do we define generalist in this sense? We just, for now, we ignore the saddles, which is very abundant in high-dimensional space. So we say if we first focus on the peaks, which are local optimum. So here, the common ones will, in some sense, represent the generalists. So they are reasonably fit in multiple environments. And the specialists are only good for a particular one. All right, so there are unique local optimum in a particular environment. So if we alternate between them, so you can imagine there are possible attractors. So one type you can easily imagine is a trap between nearby specialist peak. And the other one is a more desirable one, is a fixed point toward which the population hopefully will flow toward. So that's the generalist attractor. So the basic intuition that leads to all the later result is the variation in fitness is different between the two kinds of solutions. Right? So specialist, by definition, even if it's probably fitter than the generalist on average in a particular environment, but on average could do worse. Okay, so you see very large fitness swing as the population, as the environment switches. But in contrast, the generalist basically stays there. Right? It's like a stationary attractor in a changing environment. Okay, so that's basically the basic intuition. So this is too much for such a late time in the day, but the only thing that's important is we can construct something that we can systematically tune the ruggedness and the correlation between environment. So two parameters, and we do it in a way that they are decoupled. So can tune one of them without affecting the other. And yeah, it's something called Anki Landscape, developed by Kaufman and Levin, to really describe antibody evolution of affinity maturation, but for a particular antigen. So it's a static environment. And yeah, and to make connection with experimental data, you can decompose a measured small landscape from the deep mutational scan study. You can decompose using some kind of Fourier transform to get each order and then compute correlations. Right? So there's a pretty apparent connection we can make with data. And you can imagine the time scale would matter. And the relation between the two environments would matter too, right? So you imagine they should be connected. So the answer, yeah, later I will show you the quantitative support of the answer. But the intuition, again, is whether too fast or too slow, a good thing? Maybe not. If you imagine this dynamic switching scenario I just told you about. Because eventually what we want to have is not an equilibrium distribution in the landscape. We want something that creates a kinetic asymmetry, meaning we want more flux toward the generalist than the reverse. All right. So can we do that? The answer is certainly positive, but we're still exploring the rules. Of course, we need to both design the landscape correlation and the time scale corresponding to it. So if we do it right, in this case, we can even read the dynamics from the topography, meaning we don't need to exhaustively run a lot of simulations, but just by characterizing some topographical relationship, can we already say something about the potential of the environments? Okay. So I will first talk about that. So that's possible under certain conditions. All right, so make some assumptions, the sweep regime. So it's basically sweeps. And then it's asexual. And it's large enough, but not too large, such that it's pretty greedy in its dynamics. And the greediness will make a unique mapping of attractors. So given a landscape, we know the division of the attractors. Okay. this is showing something that was a little surprising the first time we saw it. So I'm now plotting the fraction of shared optimum among all the local optimum as a function of the correlation between environments, meaning from anti-correlated to completely identical. Right? So you can see this increases, unsurprisingly, with the correlation. But when the ruggedness increases, you see the sharing probability increases. So that says when we use more rugged landscapes, it's more likely for the peaks to be shared. So that's not completely intuitive. So that's, I think, part of the property of the NK landscape, because so K is basically how many neighbors are coupled and contribute to the fitness contribution of a site. So that's a sparser version of spin glass. But the thing is, as the K parameter increases, it not only increases the number of the peak, but also modulates the distribution of them. So they become more evenly distributed. So that's a more important contributing factor to the increasing sharing of local optimum. But this says the epistasis can play a positive role in the sense of expanding diversity of generalist solution. And another picture I want to describe to you is about this basin. So we assume a unique division of this basin of attractions. And that allows us to quantify how does environmental cycling shrink or expand the basins. So we want them to expand. So this is a positive, very optimistic example, and it does happen. So here, it's ramified because it's high dimensional. Most of the volume is on the surface. And in the two environments, so you can see these are the isolated basins on their own, so meaning in static conditions. But if they are allowed to cycle in between, you can see the peak where the population gets stuck at can be mobilized when it's switched to a different environment and be running uphill again. So that's the idea itself is not completely new. People talk about ratchet all the time, but this is kind of a high-dimensional generalization of that idea. But the thing is, it creates an effectively continuous positive slope on this alternating landscape. And another good news is we do the system for about 10 amino acids, and for that, we only need the connected chain of basins is only 6 or 7 in total. So that means not a huge number of cycles should suffice if the time scale is done right. Okay. So basically, all this connected basin is now becoming the effective basin of the attractor, which is centered at the generalist in blue. Is that the time left?
You have a minute 30 left.
That's impossible. I'll try. I have a second part. So this is the quantification. But the thing is showing both the amount and the structure of the ruggedness matters. Okay? So the best chance, as you can imagine, you don't want them to be too similar so that the basin can be enlarged, but similar enough for generalists to exist in the first place. Right. Okay. So this is a phase diagram where we have the two types of attractors. So you see this attractive red zone along the diagonal. That's where there's the best chance to evolve fit generalists, meaning they have the strongest enlargement of basin due to the cycling. And the two corners probably is where we want to push the microbes to. So they either trap at very small basins and fit, or they could even lost their way and trapped among these specialist peaks. Okay. So the experimental measurement, by doing those Fourier transform I mentioned before, can be placed on this diagram. So we have a sense before running the experiment, what's the potential of a particular pair of environments or drugs in that sense. Okay. So let's start off my second part. Maybe I can have 1 or 2 minutes very quickly. Yeah, because it looks nice. So, so this is another way of constructing landscapes. If we know what sequences we want to evolve, or the specialist on the way toward the generalist, then we can do it by a constructive approach. It's a Gartner construction where the power controls how flat top these peaks are. So yeah, so X is the vector for antibody sequence, and you can have multiple epitopes. Epitope is just the features exposed on the antigen that can be seen by the antibody. So yeah, this is just the projection of the different solutions. They form fitness islands in a sequence space. And yeah, green and orange are specialists in each environment. And there's a common one. So here. So I just want to say the most interesting regime, or what's essential for the Bayesian enlargement, is intermediate timescale, where system is not allowed to reach equilibrium, but kept at the dynamic transient regime. So that's what this little tail represents. So they didn't have time. This is very useful to prevent the specialist to reach their preferred and topically favored distribution. Okay, so to interrupt them before they get there. And so that works. There's a peak at intermediate timescale that take advantage of this. So there's enough time for generalists to evolve, but not enough time for them to specialize again. Okay. And we can do even better by doing a chirp, meaning we start slow, so generalists can be evolved, but later speeds up, so we can purge out the specialists. Say the other. So you can see the chirp protocol can beat all this fixed frequency protocol in both the number of generalists and the success rate. So pretty much ending. So yeah, this is just showing, it's an interesting analogy that the environmental changes will warm up the specialists, but not much the generalists. So the warm thing will recollect toward the colder region in the sequence space. And one last slide. We just test this using, we went, okay, it's all fine in a computer. Can we do something with the HIV data we can get from literature? So we found people can collect the main specialists and the generalists in some individual they track long enough. So one thing interesting we found is in the real data, the specialists are really not so much correlated. And that's important for this enlargement or this asymmetry toward the generalists. If we artificially make it larger, then the success rate drops a lot. So that's my message. And this is what we're also interested in, but don't have time for. So yeah, so there's a more direct connection to this theme, which I would be happy if we can talk about it. So that's about the dynamic interface between immune cell and antigen presenting cell. And we are interested in coevolution and this learning problem and the spatially distributed structures.
Good.
Okay,
all right.
thank you.
Thanks very much.
Yeah, sorry.
So, time for a couple questions.
Thanks for a great talk. I have one question about one of the assumptions you made. So you assumed that kind of the population is greedy and always goes uphill, but often, especially these viral populations are super large and lots of mutations are entering at the same time and competing, which probably breaks detailed balance. And so do you have already an idea of how this changes the picture?
Yeah, that's something we try to avoid at the beginning. But I think, so here so the B cell population itself is relatively small. So the antibody evolution is happening in this about 1,000 of cell clusters. So if we think about that, probably it's closer to a sweep situation, even though I agree, the mutation rate of B cell is pretty high, pretty close to HIV actually. Yeah, but I think the multiple mutation or interference is certainly happening. Yeah, I think there's some indication recently about that. We haven't considered, which would be nice to include and see how this protocol fail or modify. Yeah, thank you.
Other questions? If you can ask a quick one about flu vaccines. So should I be getting a vaccine every year, or should I be chirping it throughout the year?
That's a different question. Yeah, we were thinking about something we can't get rid of, basically, easily. But for flu, we are better off. Every year, we naturally control it after a while. So I would say the less, the better.
So don't get your vaccine.
If it's necessary, no, I would. But the chirp, I think, is more about something you really need the cycles to amplify it. Yeah, so it depends. Yeah, it's the most difficult situation.
All right, thanks very much.